Hello everyone, myself Dr. Deepanjan Datta from Manipal College of Pharmaceutical Sciences, and I welcome you all in the 23rd International CRS Indian Local Chapter Conference. I thank you, each one of you, for giving me an opportunity to present one of my research work on mechanistic investigation of choline-based ionogel on transdermal delivery of vancomycin hydrochloride. So in our studies, we have tried to enhance the permeation of vancomycin hydrochloride, which is a high molecular weight drug as well as a highly water soluble drug across the intact skin and tape stripped skin. So basically, we have selected three different chemical permeation enhancers, which are oleic acid, palmitic acid, and geranic acid. And these three different chemical permeation enhancers were mixed with ionic liquids, that is choline bicarbonate. And then these three different formulations were characterized for NMR, DSC, FTIR, zeta potential, and conductivity test. Next, we have also performed skin permeation studies where we have used excised porcine ear skin, and the skin integrity was measured by using TEWL, that is transepidermal water loss, and TEAR, that is transepidermal electrical resistance. The outermost layer of the skin, that is stratum corneum, which is a barrier for almost all of the drug molecules, was actually compromised by using 21 3M scotch tapes, which were applied in a circular motion and then was tape stripped in order to remove the stratum corneum and leaving or exposing the viable epidermis. Again, the transepidermal water loss and transepidermal electrical resistance was measured, which was significantly higher, concluding that the stratum corneum was compromised. These skin samples were again sandwiched between the donor and the receptor compartment of the France diffusion cell apparatus and were treated with these three different formulations for up to 48 hours. At the end of 48 hours, the studies showed that there was an enhanced permeation of vancomycin hydrochloride across the tape stripped skin, which was treated with choline bicarbonate and geranic acid, or the mixture of choline bicarbonate and geranic acid, which is termed as cage based ionic liquids. Encouraged from the results of skin permeation studies, we then loaded this cage-based ionic liquids into a gel, which is termed as cage-based ionogel formulation. And then again, this was characterized for DSC, FTIR, and contact angle measurement to understand the hydrophilicity, hydrophobicity, and the spreading nature of the gel formulations after being applied onto the human skin. Similarly, we have also used the skin samples for skin permeation studies, and we found that the enhanced permeation of a vancomycin was achieved across the tape strip skin, which was treated with cage-based ionogel formulations. The foremost important parameter for this study was to understand the mechanism that led to an enhanced permeation of vancomycin across the tape strip skin. And for that, we performed three different studies, which are DSC, FTIR, and SEM studies. So in figure 2, we can see the representative SEM images showing the surface morphology of intact stratum corneum and tape strip skin, that is the viable epidermis, which was treated for two different time points, that is 2 hours and 12 hours. And we can see that there was a wider pores or the cracks appearing on the surface of the skin samples. Next, in figure 3, we have representative DSC thermograms and FTIR spectra, which for the skin samples, which was treated for 2 hours and 12 hours. And we see with increase in the time points, there was a significant shift in the melting point as well as the wave numbers, which was due to the extraction or the fluidizations of the skin lipids. So to conclude, yes, these were the probable mechanisms which led to enhanced permeation of vancomycin across the tape strip skin samples. Again, we have performed in vivo skin irritation studies where it was performed on live healthy SD rats, where the formulation, that is cage-based ionic liquids and cage-based ionogel formulations, were applied onto the intact skin as well as compromised skins after being treated with 10 3M scotch tapes, where the stratum corneum was compromised. And again, the skin integrity was measured using transepidermal water loss as well as mean blood flow across the skin samples using laser Doppler fluorometry. Next, the animals were sacrificed and the skin samples were harvested, cryosectioned, and was stained with hematoxylin eosin chemicals to perform the histopathological studies. So on the 14th day, we found that there was a significant reversible appearance of stratum corneum, or those were actually a loose appearance of the stratum corneum without any significant changes in the vival epidermis of the skin samples. So the prepared formulations was not having any kind of toxicity or the irritations upon being applied onto the healthy skin or being applied onto the tape stripped skin samples. To conclude, the skin permeation of a highly molecular weight drug or a highly water soluble drug, which in our case is vancomycin hydrochloride as a model drug, is possible or can be achieved across a tape stripped skin when treated with a cage based ionic gel based formulation without showing any type of or any kind of potential or irritation upon being applied onto the human skin. Thank you.